Hi, everyone, and welcome to Microbial Minutes for for February 5th, 2019. I'm Julie Wolf, the Science Communications Specialist here at the American Society for Microbiology, and we're joining today to talk about what is hot in the microbial sciences. We're going to approach this with two questions this week, as we'll see on slide 2. The first question that we're going to ask is about the gut microbiome, revisiting the same topic that we had discussed last week. In this week, we're going to ask whether the gut microbiome can actively promote health. And then the second question to look at is how can a tumor-causing virus be used for good? So diving right in with our first story on slide 3. Here we're going to use a paper that was recently published in Nature, and the take-home message from this report is that there are specific members of the gut microbiota that can prevent disease, at least in mice. And so this is asking whether our gut microbiome can be used to promote health. And what they're specifically asking is whether the gut microbiota can influence a certain population of our immune cells. So there's already been research that has shown that the different gut microbiome members can affect certain types of our white blood cells called T cells and certain subpopulations within the CD4 population of T cells. These are things that help with aspects of immunity such as mucosal immunity, things that can be regulatory to dampen perhaps immune responses. But what this group wanted to look at was a particular subset of CD8 T cells called CD8 interferon gamma expressing T cells. And these are very important for their effectiveness in fighting intracellular both infections and abnormalities. So this is an important cell subpopulation that helps to activate the cytolytic T cells, the CTLs. Those are cells that will go around and look at what is inside of cells, and they will destroy cells that are presenting antigens that suggest they are either intracellularly infected by things like viruses or intracellular bacteria, and they will also detect certain tumor antigens and destroy the cells that are displaying those. And what this group first observed, as you can see on the right-hand side, is that there was an increased frequency or concentration of these CD8 interferon gamma expressing cells within a certain part of the intestine of specific pathogen-free mice. Those are SPF mice. They have normal gut microbiota. They're screened to make sure that they don't have certain types of viruses or other pathogens that might otherwise skew results. And you can see that there is a high frequency of this cell population in the colon and in the lamina propria. If they looked at this same area within mice that have no gut microbiota, these are germ-free mice. They're birthed by cesarean section and made sure to be completely free of any potential microorganisms. They did not see that same increase or high frequency of the CD8 interferon gamma expressing cells. And so what the group wanted to ask was whether there was something within the gut microbiota that promoted the aggregation or accumulation of this particular subtype. And so instead of looking at the mouse fecal or the mouse intestinal bacteria populations, they took feces or fecal microbiota from different human donors, and they looked at the ability of human microbiota to promote this same CD8 T cell subpopulation. And what they saw was that if you look in panel C there, that all of the different donor fecal microbiota were able to increase the frequency of that population. Particularly, I believe it was B5, that they saw the highest frequency. And so this really is a nice beginning to their story. They're kind of using Koch's postulates, which is used in order to associate a particular microorganism, usually with an infectious disease. But here what they're doing is showing that a particular microbial population, whether it be a single microbe or a consortia, is associated with the presence of this particular immune cell subpopulation. They were then able to transfer this from the B5 recapitulated mice who had the fecal microbiota from the human donor and put that into those mouse feces into a germ-free mouse, a second germ-free mouse, and again recapitulate that high population of that CD8 interferon gamma expressing cell population. And they saw that it was specifically due to this consortia because if they added different types of antibiotics, as you see in panel D on the right-hand side, that basically all of those would change the consortia or the population of that microbiome, and that would decrease the high prevalence of that subpopulation. And so this was the beginning of their story. From here, they were able to culture and to sequence to identify what those microbes were. And so let's look at what they found in panel 4. On slide 4. From here, they were able to, they identified a number of different strains that were part of that consortia, but there were 11 of them that were highly correlated statistically with the presence of this CD8 interferon gamma response. 7 of those were in the Bacteroidales family. Those are bacteria that we've talked about previously as being associated with good gut health for their ability to digest certain types of fibers and produce metabolites that can promote gut health. And 4 were non-Bacteroidales family members. And this duration of good gut health, or the high increase in the population of this particular subpopulation, lasted for 6 months. So they were stable, they were engrafted, one could think. And they also saw an increase in other measures of T cell immunity. Those weren't as high as specific microbes that have been associated with those particular responses, but they were still increased in their frequency of the cell types that were in that area. Now, these bacteria were found to be mostly localized to the cecum and colon. So this is the large intestine of the mice. And they looked at the mechanism. So why does this subpopulation of T cells come to this consortia? And they saw it was a combination of a number of different things. They saw that there was an increase in chemokine secretion in the gut epithelium. So those are the molecules that will recruit this subpopulation to the area. They saw that this subpopulation was able to rapidly expand. So there was likely some sort of nutrient or stimulant within the area that allowed them to expand their cell numbers once they were recruited to the area. And they saw that the antigens presented by this particular bacterial consortia was able to mediate differentiation of the T cells, T cell receptors in various ways that allowed them to recognize some of those bacterial antigens. They hypothesized that there is also some systemic effects due to the circulation of some of those bacterial metabolites. So things that are secreted within the gut might be able to make their way into the bloodstream and have better immune responses throughout the entire system of the mouse. But they did not definitively show this. And so what is the result of having these 11 different strains and this increased CD8 interferon gamma response? So they looked at 2 of those things that I had talked about at the beginning. They looked at an intracellular infection with listeria. They gavaged mice that had listeria, that had this, what they called GF, so they're germ-free, plus 11 bacteria mix. And they saw that the mice that had this 11 strain consortium mix were able to better fight off the listeria infection. And that happened, as you can see in panels B and D, where the mice lost less weight than those mice that were not treated with bacteria, or importantly, mice that were given a 10 strain mix. So they were omitting a single member of that 11 strain consortia, and they saw that there was not the same type of effect on the immune system that helped them to both clear the bacteria so that there were fewer CFU, as you see in the bottom, and also maintain mouse weight, so there was less overall damage to the mice as measured by their ability to maintain their body weight. And they also did the same experiment looking at mice that were engrafted with adenocarcinoma cells. So this is a mouse tumor, and they're looking at the tumor response. And they saw that mice that had that GF plus 11 mix responded better to anti-PD-1 treatment. This is a treatment that is going to help to eliminate some of the anti-immune responses so that they will be activated to kill the cells, the cancer cells. And they saw that, again, not only were the mice that had this 11 strain consortium better able to fight that particular tumor and to respond better, but they were better than mice that had been given only 10 of that mixture, which I think was a really nice control. Now, you might be asking, yes, but of course, people, humans, and even mice have more complex gut microbiota than simply having a mixture of 11 different strains that are very well defined. And I was thinking this as I was reading it, and they did some very nice experiments to demonstrate that, as highlighted here in the bottom box, the effect of the 11 mix was also observed within complex microbiota colonized mice harboring specific pathogen-free, so those are some of the mice we talked about at the beginning, or donor stool. So those would be undefined, extremely undefined, and also the same context from which those different microbiota had been identified. And so while this did not actually make the mainstream media, this was quite heavily discussed on social media by different scientists who are excited by the findings. If we look on slide 5, we'll see that it was shared not only by the publication that generated this, Nature, but also by other publishers who found this to be very exciting. It was also, I should mention, shared by the company which produced this research. So this was not done at an academic research institution, but was done at a company who is hoping to eventually turn this into something that can help benefit cancer patients who are receiving some of those cancer chemotherapies or cancer biologics that might help the immune system to better fight off the tumors that they have. And so the chief executive officer of Vedanta Biosciences said that this research demonstrates that specific human microbiome-derived bacteria assembled rationally into consortia can cooperatively enhance the responses to immune checkpoint inhibitors, which support our hypothesis that modulating the gut microbiota could be a powerful tool for potentiating immune responses that help fight cancer and infection. This was shared by people who study the gut microbiome and microbiome research in general. I recommend following Elizabeth Bick if you don't, but you're interested in this space. She highlights a lot of really great articles. And this was also one of the comments that I really liked on social media was that we've moved away from a microbiome atlas to applications. So being able to define using these particular microbes to assist with this particular disease or application. So very cool story from Nature. And next we're going to move into slide 6, where we will answer the question of how can we use an oncolytic virus to specifically target tumors. And this is from Translational Medicine. The take-home message from this report is that an engineered virus specifically infects and replicates in cancer cells. So this is pretty exciting. The cancer here that we're talking about is a retinoblastoma, which affects 8,000 children worldwide annually. These are often very young children. This is a genetic disorder. It's often something that's inherited where different alleles of the parents are passed on to the child, but it can also occur during just development if a mutation accumulates within the retinoblastoma gene. There is unfortunately no very good therapy. Blindness and eye loss, eventual eye loss, are very common for children who suffer from this cancer. And so the scientists in this study wanted to look at a modified oncolytic adenovirus. So adenovirus is something that is commonly found that can be the cause of the common cold, for example, but under certain circumstances and different lineages, they are associated with causing or affecting different aspects of the cell cycle that can eventually lead to cancer. In this case, this has been a very well-defined adenovirus. It's something where the modification has eliminated the ability of the adenovirus to circulate in non-tumorigenic cells. And so as you can see, I know it's a little bit, I know that the resolution is not amazing, but I was able to find an image of the modifications of this particular VCN01 strain. And in this strain, there are a couple of modifications to help encourage its growth within those tumor cells and make sure that they don't grow within normal cells. And that includes a 24 base pair deletion in the adenovirus E1A gene. This impedes viral replication in cells that have a functional RB1. And so this means that they should not be able to, because the E1A is required for viral replication, if that RB1 is functional within the cells, it will not be able to produce progeny virion. There's also an insertion of the E2F promoter under the E1A gene this permits transcriptional control in cells with high expression of free E2F1. There are some viral receptors that have been engineered into this particular strain. That includes the Coxsackie adenovirus receptor, or CAR. This is something that is commonly expressed in many different tumor types, as well as different integrins, the AV beta 3 and the AV beta 5, which are also expressed by several cancer types. And so moving to slide 7, we will discuss just some of their results. They, of course, first looked at in vitro culture, looking at the tumor cells that were cultured from different patients, and they were able to see that 11 of those different cultures were infected and killed by this VCN01 viral strain. They then did mouse experiments looking at mice who were engrafted with some of these RB tumors. And they noted that the VCN01 stopped the spread and delayed the eye removal. So if we look over on the right-hand side here, you can see that the mice that were treated with the virus, which are in blue, they did suffer from having eye removal. So they weren't able to save the eye completely, but they were able to prolong. And in some cases, quite a bit, prolong the eye use, not only its preservation within the face, but of course its function in being able to see. And so from here, they moved into 2 young patients. And this is why this is quite an exciting paper. So this had 2 patients. This is not a clinical trial, but just to look at the efficacy. Now, this is a clinically verified, clinical grade virus. So the safety within patients is already well established. And here they injected into the eye, and they saw that the virus replicated in tumor, but not in healthy tissue, which is exactly what one would hope for, so that it's specifically targeting the tumor cells within the eyes of these patients that have the retinoblastoma tumors. They did see that one tumor shrank in one of the 2 patients. These are 2 very young girls. They're 2-year-old girls. But they did not see a difference in the retinal tumors in both patients. So you can see in the lower panel here on the right-hand side that the tumor size did not actually shrink. And when they looked at the efficacy, although the tumor cells were able to have propagation of that virus, the virus did not completely kill the tumor cells, which was a shame. However, as a very preliminary trial, you can, in slide 8, you can see that this was shared by a number of different people. It wasn't picked up by a lot of places in the mainstream media, but it was shared by people who work in the medical field, shared by patent attorneys, and shared by a former Surgeon General. So Dr. Ali Khan shared this because he was excited at the unsuccessful but safe use of this particular virus. And so I think that what this does is to build a strong foundation for a tool that we might be able to use in the near future, not even in the far off future, but in the near future, in order to attack some of these types of tumors within children. There was a coverage of this on the Nature podcast. So if you want to listen to that, we'll have a link down below. And I found one article. It was actually from The Conversation, which is a place where many different experts can go to opine about a particular area. And this one was picked up. So the Conversation will publish it, and then it can be picked up by other types of outlets. And here it was picked up by San Francisco Chronicle, where the person who wrote the piece is a scientist, and she works on different types of eye diseases. And so she writes from her point of view the types of diseases that she's seen. And if you want to read a little more into her personal perspective, we'll have a link down below to that as well. Finally, I wanted to point out the similarity of using an engineered virus that has been modified in a way that will only attack very particular types of tumor subsets of cells to something that we talked about in a previous Microbial Minutes, where we talked about how Zika has been modified to treat the deadly brain cancer called glioblastoma, and that is something that you can see in the archives of our Microbial Minutes. We'll have a link down to the press release about that particular article down below as well. So that's going to bring us to our conclusion, slide 9, where we'll summarize what did we talk about in the last couple of minutes. We learned today that there are specific gut microbiota members that can prevent disease in mice. And we also talked about how an engineered virus can specifically infect and replicate in cancer cells. Now, As you can see, there were only 2 stories that we discussed, and that's because we're coming to you weekly now. If you want to catch next week's session live, go ahead and subscribe, and you'll get a notification when we're about to, a day or 2 in advance, or pop in anytime during the week. These are recorded. Go ahead and leave a note if there's a particular study that's published this week that you want us to discuss in next week's Microbial Minutes. I'm Julie Wolf. Thanks for listening.